All right, so antimicrobial susceptibility test. So for this lab, there are lots of different tests that you could do for antimicrobial susceptibility, and I'll show you a couple others in this presentation. So again, just like with endospore staining and acid-fast staining, there were particular methods that we used for those particular staining procedures. There was a particular method that we followed for this particular antibiotic sensitivity testing we did, and it's called the Kirby-Bauer method. So it's important to know that we did that particular method. We're going to do standard plate count, or pretend to do standard plate count, right? We're going to play around, and we're going to look at the results from the one that I set up. So some important terminology as it relates to antimicrobials. Antibiotics, that term a lot of times is inaccurately used. An antibiotic, the true definition of it, is a natural antimicrobial agent produced by microorganisms. So in the case of this picture here, you have our good friend penicillium, right? This mold, penicillium, that produces under certain circumstances the antibiotic that we know as penicillin. When it produces it on this agar plate, right, it diffuses out just like our discs that had antibiotics impregnated in them, and it diffused out into the agar. And in this case, it stopped the growth of the staph, the coccus that was growing on this plate, because the way that penicillin works. So these guys are fighting chemically, have been doing it for eons. We've just basically, when we use antibiotics, we've stolen their technology, right? We've taken substances that microbes are already producing to fight amongst themselves, and we're taking and using it for our own benefit. Is that the only thing we do? Do we just take chemicals from other microbes to fight microbes? No. Some of them have been chemically synthesized in laboratories, right? Some of them come from other things like plants, right? And even potentially some animal sources. So antimicrobials or antimicrobiotics is a general term, right? For anything, any substance, both natural and synthetic, right? So it includes antibiotics. But when you say antibiotic, it should be a natural substance only from a microorganism. Where if you say antimicrobial, it includes antibiotics, right? That's the safe thing to say. And so we have, substances out there that can interfere with lots of different types of microorganisms, including viruses. And some of them will kill, and some will inhibit, right? Some antibiotics only inhibit, right? They do not kill the organism. But we have an immune system that helps protect us, right? So that's what will actually kill it, right? The inhibitory ones help us in slowing it down, giving our immune system a better fighting chance. People who have compromised immune systems, though, those types of drugs wouldn't do them any good, right? The ones that just slow them down. So we have some terminology as it relates to that difference of killing as opposed to inhibiting. So I have a cute little cartoon here, right? Bacterial static are ones that, yep, it's this damn thing on my computer that I need to get removed. Stupid malware. My poor computer's infected right now. Probably why it's behaving badly. Okay, so bacterial static, right? They just slow them down. So you notice they got them tied up, right? Bactericidal, on the other hand, and of course we're being very specific here in talking about affecting only bacteria, right? This will actually kill. So some examples of some of the antibiotics that we have on the plates that we use today for bacterial static are chloramphenicol and tetracycline. Bactericidal, there's a whole class of drugs known as beta-lactam drugs. They have this similar structure to them. And penicillin and penicillin derivatives. Penicillin is one of those natural substances that chemists had then modified, right? So it's semi-synthetic, right? It's not completely synthetic, right? But it was a natural substance that we've modified. So ampicillin, which is our example of a modified penicillin, right? A lot of the psyllin drugs, the drugs that end in psyllin, are modified penicillins. So amoxicillin, right? That's a modified penicillin. And because of that, they work a little bit differently than just straight up penicillin. And that's one of the main reasons why they modified it. But all those drugs have a similar structure to them, and so we'll look at that chemical structure, what we call a beta-lactam ring structure. Aminoglycosides is a whole another grouping of bactericidal drugs. One that falls in that particular group that we use was streptomycin. And ciprofloxacin, commonly referred to just as Cipro, is another bactericidal. We had some new antibiotics that we had gotten in, and so the SXT is actually 2 drugs on that disc. Both of them by themselves are bacterial static, so they would inhibit. But when you use them in combination, they are bactericidal, right? So they will actually kill. And so drug interactions matter too. And most women are aware of this problem too as it relates to antibiotics and birth control pills, right? Antibiotics can decrease or eliminate the effectiveness of birth control. One of my nieces, I have to thank for that, right? So, you know, drug interactions can be good. In the case of 2 that are inhibitory become a killer, or you could have where they cancel each other out, right, where they're antagonistic in the case of antibiotics and birth control. So how do they work, though? Why do they inhibit or why do they kill, right? So you need to know the general ways these work, right? I have the specifications here for each one of the antibiotics. By no means do you need to memorize that, right? So for a cell, maintaining a membrane is really important, right? So if we interfere with its membrane, we could kill the cell. Its cell wall, especially for bacteria, it's made out of a specific substance that we don't have, so we can interfere with their ability to make that cell wall. We can interfere with DNA replication, right? The enzymes that catalyze that reaction. We can interfere with going from DNA to RNA transcription, right, with the enzymes that catalyze that reaction and really harm cells. We can interfere with RNA to protein translation, right, and really harm them by them not being able to make the proteins that are catalyze the reactions. We can affect specific metabolic reactions, right, specific pathways with certain drugs. So these are the main targets, right, and an antibiotic typically only targets one thing, right, it's interfering with one thing. So in the case of chloramphenicol, right, it interferes with protein synthesis. It actually prevents the peptide, that bond that forms between the amino acids when the ribosomes are putting together those pieces. Cipro interferes with the enzymes necessary for DNA replication, right? So it harms the cells, stops them from being able to grow by stopping DNA replication. For penicillin, it interferes with the linking up of the layers of sugar with the amino acids that link them together in the peptidoglycan, right? So they affect cell wall synthesis. So that's another reason why for our lab we did 2 different organisms. We did gram-positive and a gram-negative, right? Because structurally, especially as it applies to the cell wall, they're distinctly different, right? So remember, our gram-positives have that very thick layer, peptidoglycan, and the gram-negatives have a very thin layer. So they're affected differently. So if you guys notice on your plates, E. coli, did penicillin affect it? No. If you look on your plates, right, it grew all the way up to the penicillin disc. Penicillin didn't affect it at all, right? And the main reason for that, though, is that this outer membrane, penicillin, the natural penicillin, non-modified, cannot get through that outer membrane. All of these substances have to get inside to interfere, right, with the processes. So this, for gram-negatives, they're naturally resistant because of that outer membrane to penicillin, right? It can't get in. And plus, right, it's not that effective against it because, you know, they don't just have a cell wall. But the main problem with that one is that outer membrane stops it from getting in. streptomycin, skipping ahead to and tetracycline, both interfere with the ribosomes. Again, that's going to do protein synthesis. In this case, they specifically tell you, right, it binds to the 30S ribosome. This one specifically to the 16S ribosomal RNA of the small 30S ribosome, right? So specifically where it's binding to that actual ribosome and interfering with protein synthesis. Ampicillin, as I said, is a modified penicillin, and so this one can actually enter in past that outer membrane of the gram-negatives, and you'll see that it was sensitive to ampicillin on our plates, right? If you see there you actually had a zone, you're actually able to measure it. It's within the sensitive range. We're going to use E. coli and ampicillin later on in the semester. This is one of the main reasons that I make sure that we do it in this procedure, so that you guys can see that normal E. coli is sensitive to ampicillin. We, on the other hand, are going to make in this lab ampicillin-resistant E. coli. Okay. Sulfa drugs, there's a whole group of drugs, and unfortunately penicillin and sulfa drugs, there's a lot of people that are allergic to these types of drugs. It's a whole different problem, right? That's where the immune system is recognizing as something foreign and harmful, and the immune system is attacking it, that particular medication that you're taking. Sulfa drugs interfere with a particular metabolic pathway that we don't have. It's the formation of folic acid, which is needed for your B9 synthesis, which is necessary to make the enzymes for DNA replication, right? So we interfere with sulfa drugs with that folic acid synthesis. We don't do that. We have to acquire folic acid and vitamins like that from our environment, either from the things that you eat or the microbes in your gut that are making it and you absorb it. So we don't have that metabolic pathway, so it doesn't affect us. Sulfa drugs don't affect us in our metabolism of our DNA. Make sense? Right? The important thing here, too, is that we're targeting just the bacteria and not us. Another reason why the ones that affect protein synthesis don't affect us is that our ribosomes are different than bacterial ribosomes, right? So these drugs are binding to stuff that's different, right? So how many of you got this in your medicine cabinet at home? Probably most of us, right? You may even spend the extra money for the Neosporin, right? But that's all you're doing is spending extra money, right? But it is a triple antibiotic ointment. What does that mean? It has how many antibiotics? Three. And guess what? 3 different targets. So what are the chances that when you're putting this on a wound that they're resistant to all 3 different of these drugs? Very slim, right? That's the main reason why it's got 3. And they have 3 different actions. So we have polymyxin B that interferes with plasma membranes. And this is the one why it's a topical treatment and you wouldn't take it internally. It would be toxic to even your membranes. Externally, small concentrations, fine, right? You're not going to slather half your arm in this either, right? This is for minor wounds, right? Minor situations. The other is bacitracin, and again, this one, when taken internally, could be toxic. Just not because we don't have cell membranes, just the drug itself, right, can be toxic in other ways for our cells. And then the last one is the neomycin, which is where Neosporin gets its name, and it interferes with protein synthesis, right? So 3 different actions by 3 different antibiotics that are in your triple antibiotic ointment that you would have at home for treating minor wounds. So for external use only, right? Because, as we said, at least one of them, and in fact 2 of them, you take internally could be toxic. If swallowed, of course, you got to call poison control. If you actually read the label, right, small amount, right, don't cover large areas. Where is it? Consult a doctor if you have deep or puncture wounds, animal bites, or serious burns, right? This is for minor wounds, right, not for serious types of wounds. There it says it, minor. Cuts, scrapes, and burns.
Well, what would happen if you were to fall off your bike and you had a huge scrape up your arm and you did lather? Would it just break
if you put it on too much too often, you could get sick. I mean, I don't know how to, it wouldn't be good for your body. You know what I'm saying? Because then they're dealing with you destroying your own cells in addition to trying to deal with the healing and any potential infection, which hopefully you're preventing with the antibiotic, but you can make this, you could prolong healing too, right, by having done that.
Is it kind of like Germex, like if you're constantly using it, you're kind of washing away like all
your natural protection, yeah. Yeah, you can overclean. Okay, so resistance. How does resistance happen? Because we know resistance exists, right? We know that there are bacteria out there that are resistant. For instance, E. coli is naturally resistant. What is happening here? It actually is not even getting into the cell for penicillin, right? It's not getting past that outer membrane that your gram-negatives have. Another thing that could happen is that it could come into the cell, and then the cell kicks it right back out. Again, it has to stay in the cell and interfere with those mechanisms in order to be effective. So if the cell just basically bails it out like you bail a boat, that's not going to have any effect on it. The other thing that can happen is that it can enter into the cell and then be destroyed by enzymes present in the cell. And this is very common, unfortunately, with our penicillin-like drugs or our beta-lactam drugs that have this ring structure. There's lots of enzymes out there referred to as beta-galactanase enzymes. They break that ring. So those enzymes break that ring. They disrupt the structure of penicillin, can no longer bind to the proteins that are involved in the synthesis of the peptidoglycan, that cell wall layer, right? So they deactivate the antibiotic by having enzymes that digest it. Make sense? And that's the enzyme we're actually going to give our E. coli. We're going to give it beta-lactanase enzyme later on when we do a transformation experiment. So again, this type of bonding scheme is very similar to what you see between amino acids to form peptide bonds. And then the last way is alteration of the target. So notice it has to bind to something, right, in order to be effective. So here's our target molecule, here's the drug, it's binding, it's interfering. Here, notice that the shape has changed, so therefore the target has altered and the antibiotic can no longer bind to it. So this has happened in a lot of instances where the ribosome has slightly changed, and so therefore the drug can't bind to the ribosome anymore and therefore doesn't interfere with protein synthesis. So again, for each one of the drugs, when this happens, a cell may mutate, it may acquire DNA, it may change in any number of ways and become resistant. When that happens, due to the selective pressure of the environment, especially in an environment where there is antibiotics, the ones that are resistant are the ones that are going to survive, right? We refer to this as natural selection. Over time, it leads to what? Evolution, right? This is how we have change in populations over time. So the evolution that has happened, I'm trying to think if I have another one. Oh yeah, genotype versus phenotype. So their DNA changes, right? The DNA is what gives them these abilities, whether it be how to make that enzyme, right? How to make the ribosome different so it doesn't bind, have mechanisms to kick stuff out, right? Any one of those things, they may have acquired that DNA from someone else that mutated, or they may have mutated. Their genotype is their code in their DNA. Their phenotype is the physical expression. So that different protein they're making now, or that different ribosome, right? And so over time, as I said, this change that happens due to the selective pressure of, say, antibiotics, we end up with more resistance. So Staphylococcus aureus, what MRSA, or some people say MRSA, right? It's an acronym. It stands for Myctelism-Resistant Staphylococcus aureus. It is a particular strain, right, of Staph aureus that is resistant to mecticillin, which is a penicillin-like drug that was very commonly used for that type of infection. This bacteria, unfortunately, is very prevalent in our environment. Most of us carrying it in our noses, right? But when it infects wounds in our skin, it can cause serious problems. So we would take antibiotics to kill it, right? Well, we've used it so extensively and improperly for so long that we have a lot of the resistant strain out there in the environment. So then we switch to antibiotics, right? So in some cases, we've used vancomycin, So guess what we now have out there? Vancomycin-resistant Staphylococcus aureus. This will continue. And the unfortunate thing is bacteria are always going to be changing. Viruses are always going to be changing. Can we change and make new drugs to keep up with them? I hope so, but we may end up losing this war, right? Because we're going to run out of drugs. Right? We're not going to have anything that works against them. That's why it's important to what we have that we use it properly. So tests like the one we do today are sometimes better than just giving broad spectrum antibiotics like most doctors do, right? Where you're giving antibiotics that affect a wide range of bacteria, even your good bacteria, right? Instead of narrowing it down to one that would probably better suited. They don't run these types of tests nowadays typically is when the first 1 or 2 rounds of antibiotics don't work for an infection. Then they actually figure out what you've got and what antibiotics do we have to use now that the others aren't working. So, again, for these, although I have listed here, right, how some organisms are resistant, you don't by any means need to memorize this information, right? This is just to give you, right, real world examples of real stuff, right? So, resistance for some organisms that are resistant to chloramphenicol, it's because they don't take up the drug or they inactivate the drug, right? Other cases, right, they've altered the target as it applies to ciprofloxacin. Beta-galactanase enzymes, as I said, are very common for penicillin resistance, and especially staph. So for each one of these, right, there are organisms out there that are resistant for one reason or another. There are ones that are resistant to streptomycin either because of poor uptake or an enzyme that's encoded on a plasmid that they've acquired, right? There are plasmids out there called resistance plasmids. These are circular pieces of DNA that bacteria can trade with each other, information, right? So they can easily acquire that information. So how do we perform this test? So notice this one, no zone of inhibition, right? The antibiotic did nothing to the growth of the bacteria. Where here, we do have zones of inhibition where they did not grow, right? And so for this test, we measure that zone of inhibition in millimeters. And then we compare it to the known data chart. Again, I'm going to give you that chart for quizzing and testing, right? You just need to know how to interpret. No one has that chart memorized. Everybody looks up the data, right? So minimum inhibitory concentration, this is the lowest concentration, right, of a particular antimicrobial that inhibits the growth of the bacteria, right, outside of our bodies, right, in culture, in vitro. Minimum bacterial cytal concentration, this is the lowest concentration that will actually kill, right? Inhibitory is stopping growth, right? Where cytal refers to death or toxicity. Mean lethal dose, you'll see that term sometimes used in toxicology, right? And this can relate to lots of things. How toxic something is, a toxin, radiation, or a pathogen. And it's the dose that's required to kill half of the members of the tested population, right? That's why it's got the 50, LCD 50. Toxicity to host matters, right? We don't want to take, again, the triple antibiotic ointment orally, right, because it would be toxic to us as well. We want to not be toxic to us, but we want it to be toxic enough to the microorganisms that it affects them, right? So we have to have this balance. And again, anything in too high of a concentration, including water, right? If you keep drinking water and you don't pee, you will die from water toxicity, okay? So anything in a high enough concentration will be toxic, right, to the bacteria and then also potentially to you. So we want to minimize that, any type of interactions that could happen. So we want to give you the smallest concentration possible that will still affect the bacteria and therefore hopefully not harm you, right? So what's the least we got to give to hurt the bad guys, right, and not hurt us. So a lot of times we want to figure out that minimum inhibitory concentration. So for that, this particular test is called the E-test, the letter E test. And instead of just single discs like we use with a set concentration, these are strips that have a graduated concentration on them. So you'll notice that as the concentration decreases, you're going to get to a point where it no longer inhibits the growth of the bacteria. So where it intersects, where inhibition stops, that's your minimum inhibitory concentration, right? So for this one, it's, I can't even read that, 0.06564, right, for that particular drug, right, if they needed to know that particular information. I don't even know what TM stands for, but it's some drug. So how does the antibiotic get from the disc into the agar? Diffusion, right? It diffuses out, right? And it diffuses down, too. And that's why, although this is question number 5 maybe on your homework, that's why I cover it now, because it relates to this first question. So if you were actually to measure those plates, and I actually poured them for you guys, remember I showed you the tube that I poured it from? It was a measured amount of media. We do a measured amount for this test of 25 milliliters, which will give us the 4 millimeter deep agar for this test. And that's important for this particular test, right? Because if it was too thin, like for instance only 4 millimeters, would that affect our test? Is that going to affect diffusion if they're thinner? Yeah, because they're not going to have as far down to go, so therefore they're going to go what? Further out. So our zones are going to be unnaturally larger than they're supposed to be, and considering we're measuring these zones, right, that would be bad. So your plates were properly made, right, so we don't have to worry about those zones, right, not being accurate. And so Peter did the math, right, for this, right, but as I said, it's 25 milliliters is how much you would need for our standard size Petri dish to get that 4 millimeters deep. And if you were to take those plates, right, and actually measure how thick the agar is, it's about 4 millimeters. So the plates is supposed to be used within a specific time period after preparation and should be free of visible moisture, which wasn't really the case for ours, right? I had difficulties that morning when I was trying to pour our plates and I was trying to get rid of the moisture. So why was I trying to do that? Why would how would a moisture affect our diffusion? Would it make it diffuse more or less? Moisture will actually make it diffuse more, right? If you take a wet paper towel, put a couple drops of dye on it, that dye will really spread because it's going to go through the water. Or if you took a dry piece of paper towel and put a couple drops of dye, they'll stay right where you put them for the most part. Make sense? So the more moisture, the more diffusion, right? And so therefore we get, again, abnormalities in the size of our zones of inhibition. Right? So we don't want that. And we also don't want them too dry either, right? Which is why we want to make them fresh. So I made them fresh that morning. So the other thing that I did while you guys were taking your test is I set up the McFarland standard and then I had you guys take a look at it. I eyeballed it, but some machines, as we'll talk about later, and we have spectrophotometers in this lab. Unfortunately, they take special sized cuvettes and they don't take our culture tubes. So I'd have to sterilize those cuvettes to be able to use them, which I haven't bothered to do. So we just use, we just eyeball it because, again, we don't have to have it 100% accurate for this class. This isn't somebody's patient samples that we're doing. But once I prepare this, if I match it up good to that turbidity standard or 0.5 McFarland standard, about how many cells should that be? Anybody find that answer? 1.5 times 10 to the 8th cells per milliliter, right? So, after 30 minutes, are there still going to be only 1.5 times 10 to the 8th? Now, if you let it sit around for 30 minutes, an hour, you could potentially, E. coli can copy itself in 30 minutes, right? You could have double the amount of bacteria. And we want enough bacteria that when we swab, and if you swab properly, right, we get a complete lawn of growth. But if you have too much bacteria, would this affect our result? Yeah, the more bacteria, the more antibiotic you would need to kill them. So this test is standardized, right, so that we have approximation of a certain number of cells that we're plating per milliliter, a particular concentration. And as I said, in an actual lab, they have specially designed spectrophotometers. Instead of just eyeballing the turbidity, right, as we did, they'll actually use a machine to make sure that it's at the right concentration. So, as I said earlier, we use two different organisms, specifically a gram-negative versus a gram-positive. So we have E. coli as our negative representation and our staph as our gram-positive. And as we saw, especially with penicillin, there's a distinct difference of how these guys are affected by the antibiotics based on these structural differences that you have between these two organisms, right? So the fact that gram-positives are much more sensitive because of the thicker layer, and gram-negatives tend to be resistant because that outer membrane is hard to penetrate. Pseudomonas. What do you guys think? Gram-positive or gram-negative, the green plate? It's gram-negative. And it's resistant to a lot of antibiotics, right? There was only 2 zones you had to measure. And I didn't even do the vancomycin on there, but it would have been resistant to that one too. So it didn't even matter that I didn't put that one on there. Right? Just a few of the drugs work on that particular gram-negative. Gram-negatives are pretty scary because of that. Right? We have a lot of resistance. So then the important thing, right, and definitely when it comes for our quiz and test, you need to be able to interpret results. Right? So once you measure or know a zone, and in this case they said that for both of these antibiotics, they ended up with the same measured zone, 25 millimeters. So like, oh boy, which antibiotic are we going to give? They're both the same. Well, they're both really not the same at all, right? The zone just happened to be the same size. But does that really matter? No, because you don't compare one zone size to the other for this test. You measure the zone and you compare it to the table, right? Which of course I would provide you with. So according to the table, right, 25 for penicillin is resistant. It's got to be 29 or greater. So this particular hypothetical staphylococci, right, is resistant to penicillin. For the chloramphenicol, 25 is greater than 18, so therefore it's sensitive. That would be the antibiotic the doctor would prescribe to his patient. Make sense? Right? So we don't compare individual antibiotic zones to each other, right? We have to actually measure and compare to the table to determine susceptibility and resistance. Easy, right? Cakewalk. All right. So, how does it move? It diffuses, right? And it diffuses continuously. And so concentration-wise, too, right, where's the greatest concentration? Close to the disc, right? And as it diffuses out, it becomes less and less concentrated, right? So that zone where it stops growing, right, is our minimum inhibitory zone, right, at that particular concentration. So what you see here on our disc diffusion test, the Kirby-Bauer test that we do, right, this is your minimum inhibitory concentration. You just don't know what the concentration is, right? We don't really need it for that particular test. We just want to know if it's sensitive or resistant, right, or intermediate. For this type of test, they actually are diluting out in tubes the different concentrations of the antibiotic and exposing a particular concentration of the bacteria to it. These two tubes are shaded to indicate, of course, this is your negative control. The bacteria grew, no antibiotics. This one, the bacteria grew at 1 microgram per milliliter. So you don't, that's too low of concentration, right? 2, on the other hand, is the lowest concentration where there was no growth, right? At 2 micrograms per milliliter. So that's the minimum inhibitory concentration under those conditions for that particular test. So again, how do we interpret our results? So for streptomycin, and these are older results from a different semester. We'll look at our actual results in a moment. For streptomycin, it's got to be 15 or greater, right? And 20 is greater than 15, so therefore it's sensitive. So is it for our Staph aureus that we used and the Pseudomonas. For tetracycline, right, it's got to be 19 or greater. So E. coli is sensitive, the Staph is sensitive, the Pseudomonas is resistant, right, or intermediate. For penicillin, it's got to be 29 or greater. Notice E. coli is resistant because it's a gram-negative. Staph is sensitive, and the Pseudomonas is also resistant because it's a gram-negative. Can't get past that outer membrane. For chloramphenicol, it's got to be 18 or greater. So E. coli and Staph were sensitive, but in this case, Pseudomonas was resistant. In fact, right, it was only sensitive to the streptomycin and intermediate for the tetracycline. This is Peter's scary bout with Staph aureus. So he got a really bad poison ivy, and it got infected on his legs. And when he got back from Minnesota, he had a raging staph infection. So much so that when he went to the hospital, he had to get IV antibiotics and then had to take antibiotics afterwards. The good news is it wasn't MRSA. It wasn't Mictillin-resistant Staphylococcus aureus. But he's allergic to penicillin anyways, so they had to give him a different antibiotics. So why do we do this test? Right? We want to know if they're sensitive or not sensitive or resistant to the antibiotics. We have to perform the test properly, right? And then we measure our zones, and we use the interpretive chart to determine, right, if a large zone is in fact sensitive, if the immediate is intermediate in its sensitivity. We could use this to determine what antibiotic we would give a patient, right? We want to obviously give ones that are highly sensitive to the antibiotics as opposed to low. And then if we have no other choices but intermediate, right, that would be last resort. So for our actual results this semester, I have it on Canvas, but I also saved it to my computer. So the black bars indicate sensitivity. So for streptomycin, it's going to be 15 or greater. So you'll notice all these bars are either at or above. So Pseudomonas, which is our green one, right, is sensitive to streptomycin. So was E. coli, which is our blue bar, and then our pink bar is our Staph epidermis. Notice that Pseudomonas is only seen on here twice, right? For streptomycin and Cipro. We had no zones for any of the other antibiotics. It was resistant to all the others. For penicillin, notice again just the Staph, both E. coli and Pseudomonas, those 2 gram negatives, are resistant. Hence no bars. Staph was sensitive to all the antibiotics we tested. So notice all the pink bars are above the black bars, all above the sensitivity range. E. coli as well, except for E. coli is resistant to vancomycin and penicillin. I don't remember, did I write it on your guys' chart? What does it say for vancomycin? How does that work? Peptidoglycan synthesis. So it makes sense that the gram negatives are resistant to that. All right, so this chart, if you wanted to look at it, right, this is everybody's data averaged together from all the groups that we have and then graft. So now we have the tough stuff. So enumeration of bacteria. One of the ways that you could enumerate bacteria is to use a special slide known as a hemocytometer. It has a particular chamber etched into it, and when you put a cover slip on it, it has a specific volume. The chamber itself has etchings in it, lines drawn, so that you can use it as a grid to help count the cells. If you use tryptophan blue, you can actually determine living cells from dead cells. When we do the assay, the viable, sometimes referred to as plate counting, as viable cell counting, because only the ones that are alive are going to grow, right? In your original sample, the dead ones won't grow, so therefore you won't count them. But in this case, you could tell the difference. You would count each one of these 4 grids and average the number, and the volume of that is 0.1 microliters of that actual chamber. So that is equal to 1 times 10 to the negative 4 milliliters. Another way to directly count is to use biomass to measure like the weight of something. Another is to measure particular products. When I worked in the tuberculosis and microbiology, mycobacteria, leprosy research facility, one of the machines they used would actually measure ATP production from the samples to try and determine about how many cells were in the original sample, instead of actually under the microscope counting sample upon sample or doing several of these. Plate methods you couldn't do, especially with Mycobacteria leprae, because it won't grow in artificial culture. So you're limited to the number of ways you could figure out about how many were in a particular sample. So the drawbacks with serial dilutions and why we dilute in series we'll talk about in a moment. But you've got to wait for the cultures to grow, right? And so when our water, when we have that boil water advisory, that's what we're waiting on, y'all. We're waiting on cultures to grow, right? That's why it takes so long to get results, right? They're waiting to see what, if, and how many bacteria grow from those samples that they take. So we'll figure out dilution factors, right? If we want to know how many cells, we've got to know how much we've diluted it. Another way that you can determine possible cell numbers is to use a spectrophotometer where it measures the absorbance, right? How much light that doesn't get through a sample. It's 100% not absorptions or transmission if there's nothing in the blank. If there are cells there, not all the light gets through, and the meter is going to have a lower number reading. So you can use this versus cell counts for a particular cell. So then you could just use the spectrophotometer to figure out about how many cells you have in a particular culture. That's another way it can be done. So we won't be using the spectrophotometer today, right? And we eyeballed in the past. I've used the McFarland standard as our starting thing. Peter's been trying to do an experiment where he's trying to figure out how many cells per colony. And so he's been plating out, and we've been taking the, you'll see in this PowerPoint, taking ones from a plate with so many colonies on it and diluting it, and then having you guys dilute it, and then doing counting from that. As I said, we're not actually going to do the whole thing. So some of the things that we use for pipetting, we have these different devices here that allow you to use pipettes like these, right? Serological pipettes. I don't have one right in front of me right now. I have them in the back. For large volumes, like milliliter volumes, right? In the laboratory, we wouldn't mouth pipette. Remember I mentioned this at the beginning of the semester when we were talking about safety, how we don't put these things in our mouths. We're actually trying to phase out using those and using more of the tools that are used more commonly in the trade now, and these are micropipettors. And so these are the ones that I'll let you guys work with today and go over how they work, how their numbering system is. So there are 3 different volumes. These 2 use the yellow tips, the smaller volume tips. It's called the P20. It can go up to 20 microliters. The accuracy range is between 2 and 20, so you could go as little as 2 microliters with this pipetter. The red indicates a decimal, so if it's set at 0.15, it's 1.5 microliters. For the P20, it's accurate between 20 and 200. It fills the range between our P20 and our P1000. Our P1000 is accurate down to 200 and all the way up to 1,000 microliters, which is an entire mil. And because that's a larger volume, we use the larger tips, the blue tips. For this one, the red, right, indicates the highest volume. You actually have to add a 0. So at 100, it's actually 1,000, right? When this one's set at 20, it's actually 200. Okay, you add a 0. Where for the P200, whatever it says is what it is. No decimals, right? And no adding a 0. So you have to make sure you put the tip on the end. I did actually have one person yesterday not put the tip on the end. Yeah. So you have to put the tip on the end. You have to push it on firmly. Once you set your volume, right, by turning your little wheel here to your set volume, when you push down on the plunger, there's a natural stop, right? That's the measured stop that you need to stop at. You put it just enough into your liquid that'll be able to draw it up, and then you slowly release, and it will slowly draw up that fixed volume that you have set the pipetter at. And then when you're dispensing, you push past that stop so you can blow out all the liquid that's in the pipette. Now sometimes even that tiny little bit that sticks on the outside, especially if you're dealing with really small volumes. So in the lab, when I was doing pipetting with these pipetters, I was actually pipetting into little tiny wells on a microtiter well plate, right? So these are all like almost having little tiny test tubes, 96 of them. And I was putting only 4 microliters of my sample into each one of these. So I would actually use a chem wipe, a special paper towel that we use in laboratories, to wipe any excess off the tip before I would put it into my solution that was in my rack. You can do sometimes the accuracy, right? You don't want to put your tip all the way in, because then you're going to get a lot of fluid on the outside of your tip, and that would also be transferred. That make sense? And so I'll demonstrate that for you guys, and then I'll let you guys play around with them. So that's just the steps. So what do numbers mean? So Peter has a whole history of numbers for you guys here, and different tips and tricks for, and I meant to give this to you guys ahead of time, for the math involved. I'm going to skip over those slides, but they're here for you guys. And again, I may reference them if you come to me for help in understanding. But in the interest of time, I'm going to skip over them. And we'll get to, and there's also, you know, SI. If you're not familiar with metric, my son is doing metric right now at school, and we're having to measure in centimeters for his homework, and he keeps saying inches. I'm like, honey, no, it's centimeters. It's centimeters. It's metric. It's centimeters, right? So, yes, it's metric in lab. It's metric. So what the different abbreviations mean, and how to manipulate fractions. So I always refer to, Peter doesn't like this because it's kind of negative, but I say fractions are evil Siamese twins. They're stuck together, right? And I say evil because if you don't do the same thing to both sides, they get angry at you, right? So if you're trying to change your fraction by multiplying it, right, you have to multiply both the top and the bottom, the numerator and denominator, by that same number, right? Otherwise, it doesn't work out, right? And then we're angry because we got it wrong. Okay, so why do we do serial dilutions? Well, because if we were to start, say, if we wanted to do a particular dilution, if we started with a volume, one volume into a large volume, right, that's the problem. For some of these dilutions, we would literally have to take the volume of your desk, right, these lab desks, and put our sample into it. That's just not practical to have to deal with that large of a volume. So instead, we do it in series, where we dilute as we go, so that we can get up to a very large concentration without having to use large volumes of dilutant. Make sense? So, how do we do serial dilutions? So, we're going to take a small volume from our sample and dilute it into another volume. Right? Your book has you write your transferred amount. Next one. Sorry. This is terminology. So when we talk about the whole volume, we're talking about the transferred amount and the volume that was used to dilute, right? And this is a very important volume that you end up with. When we're talking about the individual tubes, we're going to refer to them as fold dilutions, right, or dilution of that tube, of that individual tube. Total tube dilution, or the dilution factor that we need for calculating, right, is when once we get past this initial tube, this dilution has to be taken into account with the next dilution that we do in the series, right? So for an individual dilution for your book, right, they have you do part over whole, which will actually give you a fraction, right? And a lot of people would say a 1 to 10 dilution, but this is somewhat ambiguous, right? And so what Peter and I have done, and what sometimes scientists do, is we do it as not as a fraction. And we say 10-fold dilution, right? Or a factor of 10. And in order to do that, we just flip it. So instead of having the hole at the bottom and the part at the top, we have the hole at the top and the part at the bottom. And we want to adjust it so that the part is 1, so that way you have a whole number and you're not dealing with fractions. Now there are several different ways in which you can achieve that. But when you have a fraction, you have 10 divided by 1 is 10, right? But we don't always have that nice even number of 1, right? As you guys will see in the handouts we have. So here's an example. Here's 5 mils, right, is the total volume, and they added 0.5 mils. What's this dilution? Still a 10-fold dilution, right? There's a couple of different ways you can approach this. You could take your calculator and take 50 and divide by 0.5. Guess what you'll get? 10, right? You could you can move the decimal point, right, over 1 so that you end up with 50 and 5, and 50 divided by 5 is 10, right? You can multiply it by 2, right, so that you get 1 at the bottom, and at the top you end up with what? 10, right? It's always the same, right? Here's another example, right, of a 10-fold dilution. In this case, it's 500 microliters, right? So they actually started with 450 and added 50 to get their total of 500 to give them a 10-fold dilution. The units don't matter as long as they're the same, but for this we always adjust it to milliliters because we always record the concentrations in milliliters. Even if we do our dilutions in microliters, you can convert between the two. The difference is a 1,000-fold difference, right? 1 milliliter equals 1,000 microliters. So, for our individual tubes, like for instance for this tube, they did 0.1 milliliters into 9.9. So what's our total volume? 10, right? And how much should we transfer? 0.1. So we would set this up, right? Whole over part, and that will actually give us 100. It's very common to do in factors of 10 and 100, but as you'll notice on some of your sheets for practice, to make sure you guys get the principle correct, we're not using tens. When we're using tens, and here it is calculated out, right, 100, it's good to start using exponents, right? It helps you keep track of the zeros, right? Because it's very easy to start losing track of zeros when you have 6 or 8 of them, right? Anything 3 or up, typically you should write it in exponents. And then also for our final answers, we need to make sure that we write them in scientific notation as well. So as I said, this example, right, ends up being 10 over 0.1, right? When you put it so that the bottom of your fraction is 1, it's a 100-fold dilution, which you can write in scientific notation, which would be 1. It would actually be 1.0 times 10 to the second to be accurate, right? So what about the next tube? So this tube, they're going 0.1 into 9.9. So what's that dilution? Same as the one before it, right? It's 9.9 plus 0.1, which gives you 10, divided by 0.1, which gives you, yet again, a 100-fold dilution. So they just did the same thing again for tube 2. Next tube. It's 1 into 9. So this one is what? 10. This is just a 10-fold, right? So 1 into 9 gives us our 10, right? And then scientific notation, you would write 1 times 10 to the 1. But simply a 10-fold dilution. And then 2 more 10-fold dilutions, right? 1 into 9, 1 into 9. This is the old scheme that we used to do. So for each one, right, here is a 1 to 100 dilution, 1 to 100 dilution, 1 to 10 dilution, 10-fold dilution, 10-fold dilution. This gives us, right, if we take into account all the dilutions, forget what it is, we'll see it in a minute. But let's do some more practice, okay? So if you had 2 milliliters of a sample and you put it into 18 milliliters of a sample, what would your dilution factor be? So what's your whole amount? 18 plus 2, right? 20 divided by your transferred amount, which is 2. So therefore it's 10. One of our favorite numbers. Oops, I'm going to give you the answer to the next one. Okay, so what about this one? 0.5 into 9.5. So what's your total volume? 10 divided by 0.5. So this one's going to be 100. Oh, actually, no. 20. This one's 20. So 10 divided by 0.5, right? If you multiply by 2 to make it 1 at the bottom, it actually ends up being 20. 20-fold dilution. As long as you set it up right? Do your total divided by your volume transferred, then you'll always get it right. And you can use your calculator if need be. This one you definitely will want for some of us, right, that aren't very good at dealing with fractions and whatnot, right? Just use your calculator. Okay, so now we got to take into account, though, these are just individual tube dilutions, but the final dilution of this tube, though, is a combination of the dilution to make the tube and the dilution that came before it, right, that we took a sample of. So the good thing is that when you use exponents, when you multiply exponents, what do you actually do? You add, right? So, right, if we want our dilution factor, which is what we actually need, we just add our exponents. Now the first tube, there isn't a dilution before it, right? So it's the same as the dilution to make the tube. But then after that, this tube is the combination of the dilution to make the tube and the dilution that we took it from. So we would multiply them. In this case, we add the exponents. So now this tube itself, as compared to the original culture, is 10,000 times diluted, right? 10 to the 4th. And then the next one, they did a 10-fold dilution, right? So it's 10 to the 5th. Another 10-fold dilution, so 10 to the 6th. Another 10-fold dilution, 10 to the 7th, right? You just keep going in series. But even if you were to multiply all of these, right? 2 plus 2 plus 1 plus 1 plus 1 gives you that 7, right? From the original, what is this? This is 10 to the 7th times diluted. Almost there. So let's do some examples, right? So if we did 10-fold dilutions, right? As we just saw, if tube 1 has been diluted 10 times, it's 10. Tube 2, if we take from 1 into 2, and we make a 10-fold dilution as we're doing that, tube 2 is actually from original culture a dilution factor of what? 10 times by 10 is 100, right? So the third tube is now 10 times as well. So it's 10 times 10 times 10 to get to that tube 3. Tube 1 is 10 times diluted. Tube 2 is 100 times diluted. And since we're doing another 10-fold, your last tube is 1,000 times in this example. If you did the same dilution over and over, right? It's just going to increase by a factor of 10. Well, let's do some odd numbers. So what if tube A is a 10-fold dilution, tube B is a 5, and tube C is a 2? What's your final dilution factor for tube C? A times B times C, right?
100.
Yep. So A times B times C, right? So A times B gives us 50, right? If you do, again, A times B times C, right? 10 times 5 times 2 gives us 100. Notice as I did when I was talking about our example, you don't have to keep multiplying each time, right? Once what you have what B is, you can just multiply what you did to get C to find out the final for C, right? You just keep going down the series, as long as you don't mess up, right? So what's another way you can get the same answer? You can take this and just simply divide it, times it by 2. I'm all right today, right? So just go down the series. So CFUs per milliliter, right, is what we call the titer or the concentration. How do we calculate this? When I plated these, I did not take an entire mill and put it onto those plates. Part of the reason for that will be because it's too hard to get all that spread out and absorbed onto the plates, right? It's too large of a volume for our plates. So a lot of times we're going to take a smaller volume. Do we have to take that into consideration in our calculations? Absolutely, right? Did you guys watch the video where they're filling up the different colored tubes, right? And especially when we're talking about the next one where they're just taking cells or particles, sometimes, you know, you won't even pick up all of them, right? So we have to take into consideration, because if I put an entire mill on there as opposed to just 1/10 of a mill, is there less cells? Yeah, there's considerably less cells in 1/10 of a mill as opposed to if I put the whole mill on there, right? So I have to take that volume that I actually plated into consideration. Does that make sense to you guys? So that has to be in our equation, right? So your serial dilutions until you can accurately count the cell concentration. So we dilute these two in series, right, so that hopefully, right, we get an accountable number of cells on that plate. So this is where, as I said, that volume matters. We didn't spread out all of it. So it has to be a fraction of it. So let's look at the equation. So first thing that we looked at, right, is the actual volume we plated versus how many cells we counted. But also we have to take into account the dilution of that tube, right? So our final equation that you guys need to know, right, is CFUs divided by the volume plated, then times by our dilution factor. Right? So we'll go over a couple examples. So when determining the CFUs of a given dilution, a countable plate is between 30 and 300, right? So obviously you wouldn't want to count more than 300. Less than 30, right, isn't within the statistically accurate range. That's the range I give in your book, so that's the one we're going to go by. Other laboratories use different ranges. One of the other common ones that Peter used in his lab was 20 to 200. But, you know, relatively around the same number. All right, so for this dilution scheme, and in fact, I want to switch it. Oh, I have the other one later. Okay. So, right, we would calculate individual tube dilutions as here, right? And then what the final dilution is counting in the series for each individual tube. And then we would count the colonies on the plate. And so now we have all the information we need to figure out that concentration of this original culture. So we had 120 CFUs and we plated 0.1 milliliters, which was actually measured in microliters, but that's the same as 100 microliters, of a million-fold dilution. So that's 10 to the what? What's a million? How many zeros? 6. 6 zeros. 6 zeros. So we set up our equation, right? 120 CFUs divided by 0.1 milliliters times by our dilution factor, right, which is a million, so it's 10 to the 6th. When you calculate that out, right, you can multiply these two numbers before the exponents together, just as normal. But remember, when multiplying exponents, right, we add them. So that we get a final concentration of 1.2 times 10 to the 9th CFUs per milliliter. Note that, as I said, some of our dilution schemes, right, in that packet that I gave you, right, this number isn't 1, right? It's some other number, some other factor, right? It isn't just to the power of 10. So make sure that you keep track of your numbers. So here's another example. If you plated 0.05 of a 10 to the 4th dilution and you counted 200 CFUs, what's your titer? So how would we set this up? 200 divided by 0.05 times by your dilution factor, which is 1 times 10 to the 4th. Right? Like that. So then once we had that concentration, we could actually calculate about how many CFUs, on average about how many cells. We say CFU because it stands for colony forming units, because it could have been more than a single cell that made that colony, right? So we can't say cells. We don't know for a fact that it was a cell. Okay, so we say colony forming units. So if we know that the volume of the original culture was 200 milliliters, we could figure out how many total CFUs by just multiplying by our titer, our concentration. What happens to our milliliters? They cancel out, right? And so in this case, in that original flask of about 200 milliliters, total contained about 8 times 10 to the 9. That's 8 billion potential cells in that original sample. I can tell you, it probably wasn't that cloudy. So in your book, right, their equation's a little flipped, right, because we got rid of some fractions and stuff in the beginning. So this slide just shows you a comparison between the two. The nice thing about math is that you can do things different ways, and as long as you calculate it correctly, you'll still get the same answer, right? As long as you treat those evil Siamese twins good. All right, so you guys have already measured, right? And then, as I said, you guys I'm going to show you and let you guys play around with pipetting today. We're not actually going to do the full-blown procedure with bacteria. I spoke about this earlier, and especially when you guys prepared your plates. My other class had already prepared theirs, right? The importance of actually following the procedure so that we have accurate results. So some plates, especially for Peter's class, they didn't have any bacteria growing on any of the plates. So considering we had bacteria on some of the plates, it was an issue of didn't put the bacteria on the plate, right? Or miscommunication amongst the group as to whether there was bacteria put on the plate or not. And then, of course, if you have partial growth, right, this is going to make it really hard to be able to interpret our results. So this, as I said, goes through the procedure that Peter's been working on, and it would help us calculate about how many cells are in an actual colony. So all the steps are the same as to the procedure we have, except of the starting concentration isn't set up. So this is the way he sets up his starting concentration. But this is the dilution scheme. And I want to go to microliters. Okay. So what you guys have there on your sheet is in microliters, right? Which is equivalent to the milliliters seen here. So even if you were to calculate it either way, you would get the same dilution scheme, right? That's the point that I wanted to make. So for this dilution scheme, right, see if you, I'm going to back it up, or it shows the answers. And see if you guys on that single worksheet that I have for you today, let's see if we can calculate, right, individual tube dilutions, right, dilution factor, and then I'm going to give you guys, remember I said add W because it's actually my plate 2 that ended up being countable. And my plate 2 was 69 colonies. I'm going to show you those plates in a second. So I'm going to go ahead and stop my recording for now.